Okay, we don't have a lot left in this chapter, so this should move pretty quickly. Okay, let's see. Okay, good stuff. So we're going to start here with passive versus active transport. Okay, and I want you to grasp um, the main traits of each one. So let's see. I have a tendency to run it. PowerPoints are really supposed to be a guideline, and I kind of have all this memorized. So sometimes I wonder if the PowerPoints are helping me or not. But anyways, we'll start here. We have passive transport and active transport. Okay, and the reason I have these color coded is because passive transport, by definition, means transport that does not require energy. Active transport, on the other hand, does require energy. And by energy, I mean the hydrolysis of adenosine triphosphate, this yummy little molecule over here, ATP. And let's see. In passive transport, you have simple and facilitated. They both, neither of these require energy. Another trait that is similar to both of them is that they both move from an area of high concentration to an area of low concentration. So notice that in simple diffusion, we have a lot of solute here, very little here. So simple diffusion is going down the gradient, okay? Notice that in facilitated diffusion, the same thing is taking place. We have a high concentration here, we have a low concentration here, and so we're going from an area of high concentration to an area of low concentration. That's facilitated diffusion. Active transport, well, is using energy. Anytime that you're using energy, you're doing something that Mother Nature would not do by herself. Okay, so in this instance, we are going against the current. So that means that we're going to go from an area of low concentration to an area of high concentration. Okay. All right, going back to passive transport. Okay. Simple diffusion, as the illustration demonstrates, implies that nothing is necessary for it to happen. Okay, so what are we looking at? You already know the answer to this. You're looking for small hydrophobic molecules. You're looking for some large hydrophobic molecules, and then osmosis, right? Because we are locked to those things that can make it through the phospholipid bilayer, and those are predominantly small nonpolar molecules, number one. Occasionally, some large nonpolar molecules, number two. Okay, how do we know that? Remember, hydrophobic hormones have to make it through the plasma membrane. Cholesterol can make it through. Okay, um, and then of course water. Okay, osmosis. So that's simple diffusion down the gradient. With facilitated diffusion, the passive materials is aided by both a concentration gradient, we already know that, and by a transport protein. Okay. Now, why is this? The reason is because in the example here, we're using yummy little sugar. Okay. Yummy little glucose. And as you know, yummy little glucose is polar. How do we know it's polar? Because by definition, sugar has hydroxides, and hydroxides have an oxygen. Oxygen is very electronegative, so anything that has a hydroxide on it is going to be electronegative. So sugars have a tendency to have a net negative character. That makes sugar extremely hydrophilic, water loving, okay? Which means that it's never going to go through simple diffusion, never going to happen. But if you have a specially designed channel where the outside of the channel is hydrophobic, and the inside of the channel is hydrophilic, the outside of the channel in this integral protein, right, will associate with a phospholipid bilayer, and the inside channel will associate with the hydrophilic character of the sugar and allow it to move down as gradient. So with facilitated diffusion, you always have to have that transport protein, also called a channel. Okay. In active transport, molecules move through a transport protein, but now energy must be expended to move them against their concentration gradient. Really, we've covered everything that's important about this. Okay. Okay, so you can read it with me if you want, but we already covered it, didn't we? There are two forms of passive transport, simple diffusion and facilitated diffusion. Okay. They both are the same and that they both move down their concentration gradient, but they're different in the fact that facilitated diffusion must have a transport protein or a channel protein, right? Okay, a concentration gradient is all that is required for simple diffusion to operate. Facilitated, we already know this, okay? Facilitated diffusion, here, let's read a third slide. This is the same thing. In facilitated diffusion, transport proteins function as channels for larger hydrophilic substances that, because of their size and electrical charge, cannot diffuse through the hydrophobic portion of the plasma membrane. So, if it's large and polar, you need facilitated diffusion. If it's large and nonpolar, not so much. Okay. Here we are, facilitated diffusion. We have yummy low glucose. We have our transport protein or channel. The glucose associates with the channel. Once it associates with it, it causes the channel to change shape, releasing the inside. Once it gets released, the channel changes back in shape. Active transport. Let's see. So in active transport, we need ATP. We're going from low concentration to high concentration. Chemical pumps move compounds across the plasma membrane against their concentration gradient. Okay, we know that. And then we go into a special example of active transport. Okay, and that's the sodium-potassium pump. Oh my goodness, what an incredibly important little pump. Okay. Let's see. Let's go to the picture first. Okay. So here's the outside and the inside of the cell is important. Okay. So this is the outside of the cell. This is the inside of the cell. And here we're going to start with this conformation or this shape of the sodium potassium pump, where 3 sodiums associate with the pump. Okay. Once the 3 sodiums associate with the pump from the inside of the cell, then energy, ATP, is harnessed, causing the pump to change shape. Faces to the outside, and then sodium gets released to the outside of the cell. Now, as we have said so many times, active transport means that we're moving solutes from an area of low concentration to an area of high concentration. So what this means is that there's more sodium out here than there is inside. And we're continuing to pump sodium out. Okay? So the sodium goes out. Once the sodium goes out, now 2 potassiums come in. Okay? Notice that they associate with it. And once the 2 potassiums associate with the sodium potassium pump, it changes shape, releasing the potassiums inside. And once the potassiums are released inside, it is free to obtain 3 sodiums again. So every time that the pump works, 3 sodiums are pushed out and 2 potassiums are pushed in. There's more sodium outside than there is inside, and there's more potassium inside than there is outside. Okay. And the fact that we're using ATP here, okay, and the fact that we're going against the gradient of not one, but two ions is the reason that this is active, okay? Where do we use it? Where don't we use it? So if you take anatomy and physiology, I hope some of you do, you're going to learn about the action potential of the neuron. The action potential of the neuron is dependent on sodium and potassium, okay? The way that your muscles move, that action potential goes from sodium and potassium in your nerves to the release of calcium in the sarcolemma of your muscle fibers. Very cool. In your intestine, in your small intestine, okay, we capture sugar from your food by creating a gradient with a sodium potassium pump, okay, and that's what this is talking about. So one example of such transport is the pumping of glucose into cells that line the small intestine. Here, the active transport of the sodium potassium pump maintains a high gradient of potassium inside the cell, right? Potassium inside the cell, sodium outside. This potassium gradient in turn is used to drive glucose from the lumen of the intestine into the cell against this gradient. So what happens, we have a whole bunch of potassium, okay? And this doesn't show it, but over here, down the plasma membrane, there's another channel. That channel is called an antiporter, okay? And what that antiporter does is, well, very much like this guy does, when something comes in, something must come out, okay? So because we have so much potassium inside, the antiporter will trade potassium for sugar. And the sugar is going against the gradient. Why? Because this is the food that you've eaten in here. There's a little bit of sugar here, and this greedy little cell is like, no, we need that sugar. So we're gonna, even if there's a little bit of sugar, we're still gonna pick it up, okay? But in order to do that, in order to drive sugar against this gradient, we have to push out with the gradient. So potassium goes out and glucose comes in. And we capture that glucose, which we use as energy. Or in my case, fat. Is that terrible? Okay. So notice the use of the ATP in the second step. This makes the sodium potassium pump active transport. Okay, moving larger substances. So we have exocytosis, okay, and endocytosis. Both mechanisms employ vesicles, which are membrane-lined enclosures that alternately bud off from membranes or fuse with them. Okay, and here we go. So here we have the author's illustration, and here we have an electron micrograph showing the same thing. Okay, so this is exocytosis, and you have a transport vesicle, and this is not happening randomly, right? You should know by now this is not happening randomly. This vesicle is tethered to the endoskeleton of the cell, and right here we have a microtubule running over here, and we have a motor protein on that microtubule, and that microtubule has a good handle on this transport vesicle and is moving it along, and is going to get rid of this, whatever this happens to be. It might be a hormone, it might be an essential protein, it might be waste, it's whatever, right? And here it is happening in real life. Very, very cool. So this is exocytosis. Exo means out to the outside of the cell, okay? So we're moving something from the inside to the outside. Endocytosis, there are two principal forms of endocytosis. One is pinocytosis and phagocytosis. Pinocytosis is the movement of small molecules into the cell. So we call it cell drinking. So this is cell drinking, and this is cell eating, Okay, so in pinocytosis is the movement of moderately sized molecules into the cell. Okay, phagocytosis, cell eating, is when certain cells use pseudopodia or false feet to surround and engulf whole cells. Uh, Remember, we're showing that renal monocyte going after that poor defenseless E. coli last chapter, and it was using amoeboid motion to get there, and the actin filaments were surrounding the E. coli to consume it. That is phagocytosis. Why? Because an E. coli is a big piece of something, so you're obviously not going to drink it, you're going to eat it, right? And so here's pinocytosis, and this is actually a specific type of pinocytosis. This is called receptor-mediated endocytosis, okay? And receptor-mediated endocytosis is still endocytosis, is still pinocytosis. So endo, meaning that we're moving something into the cell, is still pinocytosis because the molecules that are coming in are small, okay? But now we have this receptor to this clathrin-covered pit, and what's significant about this is that what the cell is drinking is a specific molecule. Why? Because that receptor is being very specific for a singular type of molecule. The cell needs one thing and is being very, very specific. Okay. So it says in this form of pinocytosis, cell drinking, called clathrin-mediated or receptor-mediated endocytosis, cell surface receptors bind to individual molecules of the substance to be taken into the cell, and then move laterally across the plasma membrane to a pit coated on its underside with a protein clathrin. That will become a vesicle that moves into the cell. So this is a very specific type of cell drinking or pinocytosis. And again, this is called receptor-mediated endocytosis. And that's when the cell chooses to be very specific about what it's going to take in. Here's phagocytosis. Okay, here's a monocyte consuming a yeast cell. It looks like that yeast cell has a face. And here's the phagocytosis of some kind of a bacterium via pseudopodia. That's phagocytosis. And believe it or not, that's the end of it. I hope I didn't bore you guys, but this is supposed to be a fun chapter. I don't know about you guys, but I'm going to see if I can't make something like that. Virgin, of course, right? Virgin. Tomorrow's a school night. You can't be. You can drink colored drinks. So just virgin colored drinks. Okay. Have a wonderful day. I'll see you guys later.